So good afternoon, everybody. On behalf of the Masonic Cancer Center, I'd like to welcome you to the 2025 Kersey Memorial Lectureship. I just want to say when I met John first, he was starting to plan the Masonic Cancer Center. I was young and naive at that time. I never thought I would be sitting in his shoes and really having the honor to introduce this lectureship on his behalf. It's wonderful to see so many colleagues, trainees, and friends gathered to honor the remarkable legacy of Dr. John Kersey. I'd like to give a special heartfelt thanks to Ann Kersey. Ann, I saw you in the lobby. If you stand up and wave at least to let us know who you are. Her continued efforts really help support Dr. Kersey's legacy through this annual lectureship. I also want to note following today's presentations that we will be offering refreshments in the lobby, so please help us join in the celebration. To begin our program, it is really my great pleasure to introduce my friend, colleague, and many times an advisor, Dr. Tucker Labine, Professor Emeritus and longtime colleague of Dr. Kersey's. Tucker worked closely with John during the formative years of the Developing Cancer Center as the founding deputy director, an honorable distinction, Tucker. He also played a pivotal role in forming the Center for Immunology and held roles as vice dean in the medical school, vice dean for research in the medical school, and associate VP for research in the academic health center. So please help me joining. Tucker, why don't you come to the podium? Thank you very much for being here with us today.
So what a great opportunity for me to make a few comments about my extraordinary friend, John Kersey. So I'm going to start with a bit of an odd introduction. All of you here at one time or another have heard the phrase, a man walks into a bar. And then there's some subsequent story. Well, I have a personal story about a man walking into a bar, and that was me. 50 years ago, this month, I was attending a small meeting of the American Society of Microbiology in Fargo, North Dakota. And there was a speaker, it turned out a very young John Kersey, who was giving a talk on childhood leukemia and subsets of T lymphocytes, which at the time we only described as helpers and suppressors because that's the degree of knowledge that we had. I was quite taken by this presentation, not only by the questions that John was addressing, but his enthusiasm and optimism about the science. So the talk ends, the day ends, at a Holiday Inn, believe it or not, in Fargo, North Dakota. And I'm walking out of the hotel, and I look in the bar, and there is John with his host for the day having a beverage or something before he came back to Twin Cities. And I paused, literally, at that door and wondered do I have the courage to go in there and introduce myself? Because, as the contemporary phrase goes, I had a very severe case of lack of confidence. So instead of leaving the building, I went in, spent 45 minutes or an hour with John, and at the end of the little discussion, he said, you know, when you're finished with your training, you have to come to Minneapolis and do a postdoc with me. I came in August 1977. I walked away in January of 2022. And I can tell you, as I have stated many times, that decision changed my life, big time. So that's what makes it all the more special to say just a few other things here about John. As many of you know, he spent his entire career here, with the exception of 2 small sabbaticals, one at the Imperial Cancer Research Fund in London with Mel Greaves, and the other at the National Cancer Institute with Stan Korsmeyer. John's lifelong interests were at the interface between bone marrow transplantation and childhood leukemia. I think his real scientific passion toward the end in particular was elucidating the molecular drivers of a rare type of leukemia called infant ALL, that has this interesting 411 translocation encoding the MLLAF4 transgene. And in the context of his interest in molecular drivers, I can make the case that, like our guest speaker today, who is an interest and made major contributions to molecular drivers of renal cell carcinoma, they knew what they were doing and they were ahead of their time. So in 1990 or so, John was asked by then Dean David Brown to form a small committee to look over the cancer research landscape at the university, which at the time was just splintered and fractured. None of us really knew who was doing what beyond our little immediate groups. And John took that on with the vision and the commitment from the first day that he was going to build out a program that could compete to become a National Cancer Institute Comprehensive Cancer Center. And 8 years later, we were awarded an NCI Comprehensive Cancer Center designation. And I would say through no small role of Mary Sumpman, who's sitting next to Ann Kersey. That was a remarkable accomplishment in a large, complex public university, and I think it still stands as John's greatest accomplishment. So finally, for those who didn't know him, he was a remarkable communicator who always put you first when you were in his presence. It was always about you. And if you went into his office and had an ask for space or money or something, and he couldn't accommodate your ask, most of the time you left his office still feeling like you learned something, and some people even thought that they still won. So that's an attribute that I've rarely seen in my career. So I'm going to close by directing my comments to Jeff for a moment. Jeff, you've stepped into this role. John held the role, I think, for about 16 years. Our great friend Doug Yee held it for about 16 years. So I'm just doing some simple math to 2042. Thank you.
No way, Tucker, no way. So thanks, Tucker, for really highlighting John's lasting contributions to this institution and to cancer research globally. It is now my distinct honor to introduce an extraordinary scientist and leader to our 2025 Kersey Memorial Lecturer, Dr. Kimryn Rathmell. Dr. Rathmell is a distinguished physician scientist, educator, and leader whose work has significantly shaped the field of cancer research and care. She began her journey in medicine and science with a bachelor's degree in biology and biochemistry from the University of Northern Iowa. She then earned her MD and PhD in biophysics at Stanford University, followed by training in internal medicine, medical oncology, and postdoctoral research at the University of Pennsylvania and University of North Carolina at Chapel Hill. Dr. Rathmell is widely recognized in her pioneering work, as we already heard, in understanding the molecular and genetic drivers of kidney cancers. Her laboratory has advanced our knowledge of how these tumors develop and has helped translate these insights into improving patient care. In the recent years, she has had some of the most consequential leadership roles in academic medicine and cancer research. She served as the director of the National Cancer Institute from 2023 to 2025, where she really helped shape the national agenda for cancer research and innovation. In May of 2025, she took the role of Chief Executive Officer of the James Cancer Hospital at The Ohio State University. What sets Dr. Rathmell apart is not only her scientific and clinical excellence, but her commitment to bridging discovery and care, to mentoring others, and to ensuring that our fight against cancer leaves no one behind. Her leadership exemplifies how discovery, clinical care, and policy can come together to achieve transformative impact, and this was really a vision shared by John Kersey. So Kim, really come to the stage and welcome to the University of Minnesota.
Thanks. All right, well, thank you very much. I already told you a little bit about what we're going to talk about. We're going to talk some about metabolism and metastasis. But then I think it's worth talking about sort of where we are in medical science. I do have this sort of luxury of, or privilege, I don't know, of having been in lots of different places. I feel a little bit Forrest Gump-like, right? Like I've got to be in lots of different rooms as a lot of changes are happening. And so it's stuff that we should talk about because this is stuff that's very, very much real. Okay, but let's talk with science. Science is easiest to talk about. So my lab has been very interested in metabolism for a long time. You know, that comes a little bit from the fact that I study kidney cancer, and metabolic shifts are really, really common in kidney cancer. And this has been a part of cancer for a long time. Over 100 years, we've known about the Warburg phenomenon, which is characterized by shifts in metabolism where where cancers prefer to use glucose as a dominant energy source. And this is the basis of lots of things that we do, how we talk about cancer sometimes, but also very clinically relevant products that come from this. For example, PET imaging. So PET imaging uses fluorodeoxyglucose. Ooh, that came through all grimy, but you get the gist. So fluorodeoxyglucose goes in through the same transporters as glucose, but can't get further metabolized. It gets trapped in the cell. It's fluoridated, it's radioactive, and so you can see a spot where there are cells that are more actively taking up glucose, right? That gives us the beautiful images that we use all the time to try to characterize tumors that are present in many different kinds of cancers. So my laboratory was really interested in this for a couple of different reasons. Number one is we were doing a bunch of PET imaging, and it didn't always match. So I study von Hippel-Lindau mutated kidney cancers, and when you have a VHL mutation, that causes upregulation of a protein called HIF, hypoxia-inducible factor, in cells in the dish. That always causes an upregulation of glucose transporters, all of the enzymes of glycolysis. It's a perfect storm for Warburg metabolism. Okay, but anyone here who has ever ordered a PET scan on a kidney cancer patient will tell you, but they're not very active on PET scan. And it's not totally true that they're inactive. The primary tumors tend to be inactive, but the metastases tend to be more positive on a PET scan. And we were doing a lot of kind of tissue correlates, trying to understand what was going on, getting ourselves really frustrated, until a graduate student, there are two students here, Brad Reinfeld on the left and Matt Madden on the right, went back to the original papers of Warburg to say, how did Warburg come up with this concept? And it turns out Warburg came up with this concept not by looking at individual cancer cells, but looking at the whole tumor and looking at the effect of metabolism that came from a whole tumor chunk, literally a slice of tumor. And that's what's shown there. So we started asking, like, what's really going on? The reason there's two students who were involved in this, one of those was mine, that's Brad. So we were all hardcore tumor cell metabolism. And Matt, who worked in my husband's lab, many in this audience know my husband. He's an immunologist, very out at front on immunometabolism. And so Matt was studying T-cell metabolism and T-cell utilization of glucose. And Matt and Brad were hanging out together in the pandemic, as you can tell, talking about whose cells would use more glucose. And in fact, if they were both really dependent on glucose, then there shouldn't be enough glucose present in the media anymore, that there should be some competition that was eating it all up. Sorry, I've given it away. T cells use glucose too. If you do a PET scan for things like a patient who just had a COVID injection, their deltoid will light up because of immune cells, but you can also see PET positivity with rheumatoid arthritis. So glucose goes to lots of things, not just cancer cells. And so we did the initial experiment, which was we called it the tumor juice experiment, which was we took tumors, kidney tumors, because that's what we study, and we looked at interstitial fluid. Our assumption was going to be that glucose was going to be really low, and we were going to be looking at competition. But instead what we found, and what you can see in the volcano plot on the right, was that actually there's plenty of glucose in the interstitial fluid. It's roughly at the same amount that there is in corresponding normal kidney. So there may be micro neighborhoods of low glucose, but on average, homeostasis works, and there's plenty of glucose around. That didn't really deter them, though. They still really wanted to know whose cells would win to take up the glucose. And so the only way to do that was to inject mice with FDG, like you're doing a PET scan. In fact, that's a PET scan of a mouse with an implanted tumor from a syngeneic model system, so intact immune system. And then take out that tumor, separate all the cells very carefully. We used a magnet to do that, and then count radioactivity. A very simple, it was actually quite labor intensive, but simple experiment. And here's what we found. So I was betting on tumors because I knew what the mutations were in those tumors. Jeff and Matt were betting on T cells. And we looked at a bunch of different model systems, and these are the different graduate students who volunteered their model systems. And what you can see with the red bar is that in every single case, the immune cells would win, okay? We looked at whole tumor, cancer cell was on average comparable to the whole tumor, and then when we looked at immune cells, just CD45 positive, they were always higher. Okay, so it looked like Jeff was gonna win. And, I pulled the punchline out, sorry. The punchline was that actually it was not the T cells either, it was the macrophages. But what we showed in that whole series of experiments was that the cells that are present in the tumor utilize very different metabolic processes. Basically, they choose what they're gonna eat for breakfast. They don't just eat whatever is out there, they have a hardwired program that tells them what they wanna use. The glucose was highly skewed toward going to macrophages. T cells and cancer cells liked lipids, they liked proteins. So you have to think about what you're gonna choose, just like this kid. Okay, so the follow-up to that was a whole lot of different questions, and this is just one of them. So Emily Arner's was a postdoc in the laboratory, and she was very curious, again, back to the cancer cells. What was it about these hardwired programs that was different between those primary tumors and the metastatic tumors? We have a whole thing looking at macrophages as well, but we were really curious about the dynamic shifts in tumors that might be present as you move from hanging out as a primary tumor to moving all the way over to become a metastasis. We know that it's an energy-dependent process. We know that tumors become much more PET avid. Some of that might be infiltration of macrophages, but we think there might actually be something that's happening in the tumor cells as well. And so she decided to try to unravel that. She did that by, again, taking some tumor model systems and looking for the expression of metabolic gene signature, or genes, that correlated with metastasis. Some that correlated with the occurrence of metastasis, but in this example, these are correlated with expression of EMT-type genes. There's a lot of genes in there, but one with just one red dot was where we decided to put our focus. A protein called NDUF-A4L2. It's a mouthful. Been characterized before, but not a lot of literature on NDUF-A4L2. It's present in the mitochondria. We picked it because it's downstream of HIF, very interesting. So why would it be more on in the metastasis cells than in the non-metastatic? I still don't know. But we knew that it had a connection that's going to be very closely tied to kidney cancer if it's a HIF target. And it was suspected that it would act as a regulator of mitochondria, electron transport complex 1. So dive back into the literature. It turns out it's been seen before. So it's been seen actually several years ago, a paper in 2016 that looked at kidney cancer and showed that NDUF-A4L2, in fact, in primary kidney tumors that don't have evidence of metastasis, if they have high levels of NDUF-A4L2, they do much worse. Okay. We showed this as well in more modern data sets, but this is across the board. If you have high levels of NDUF-A4L2, you are more likely to develop metastatic disease or more likely to be metastatic. So we played with NDUF-A4L2 in our cell lines. It was highly expressed to begin with in our more metastatically avid or capable tumors. So we knocked it down and then we put it back on again and showed that in fact it does hit electron transport complex 1. So probably the easiest thing to look at is the seahorse assay, where you can see in the black line, that's baseline, low levels of mitochondrial activity or oxidative phosphorylation. We knock it out, they pop right back up, they're very happy to go. You overexpress it and they go right back down. So it's like a switch for the electron transport chain. But what it is a switch that goes a lot further than that. And this is just a bunch of data kind of all in one to kind of kickstart so I can get onto the second half of the talk. But what we showed is that it's not just changing the metabolic activity of those cells, but it actually informs the expression of E-cadherin. So you can see that in the high baseline expression, very low levels of E-cadherin, when we knock it down, E-cadherin pops up. Cells actually migrate less. We get less metastasis in this metastasis model. And when we rescue it, E-cadherin goes down, and the cells metastasize again. They and are much more migratory. So why would metabolism be tied to EMT or some metastatic proclivity? Well, we think that it's actually a connection that's tied to epigenetic changes. So what we can see here, and this is well known, that if you change levels of succinate or alpha-ketoglutarate, you can actually highly influence histone methylation, and that will change your gene expression. So when you change electron transport, you change succinate. So you can see knockout, succinate goes up, alpha-ketoglutarate goes down. When you re-express, the reverse happens, as you might expect. And there's also a downstream effect that's hitting, here we're looking at histone methylation in the gene E-cadherin. And so why E-cadherin is turning off and then on is because of changes in the metabolic activity of these cells, but it's really a signaling pathway that's important for whether those cells are capable of metastasis or not. So really an interesting tie-in, and shifts the model from this energy-dependent focus, although that's not unimportant, but that there's actually a whole lot more than the effect on oxphos, but downstream of that is things like succinate, and alpha-ketoglutarate can change your epigenetic profile and lead to a dynamic shift in metabolism. So that's the science in the talk. Just a quick look at the former laboratory. My new lab has 3 people in it, and couldn't do this without all of this work. But here's the thing that I also really want to talk about, which is that just like these cells, right? We're living in a bit of a phase shift right now. The question is this a phase shift that's like EMT, right? That we're going to go T for a little while, or we'll come back, or T is the transition. We're going to go mesenchymal and then epithelial and back. Basically, is it reversible, right? Some of you are probably physicists at heart, right? Is this a phase shift where it's like parallel universe? I will say it feels like that sometimes. But this is the analogy that I like most, especially when I'm talking to a medical audience, okay? I don't know if we have cardiologists in the room. I know it's always dangerous because I'm an oncologist. So oncologists are notoriously bad cardiologists. No cardiologists? Are you? Okay. So I'm going to read an EKG like an oncologist. So this EKG, which is already well labeled, okay, so we don't have to work too hard at it, is a patient who starts out in ventricular tachycardia. Okay, it's a slow VTach, as you can see. And how do you get yourself out of ventricular tachycardia? Okay, on TV, you run in with paddles and you shock, right? Okay, that's one, the flashy way. But if you have a pacemaker in, what actually happens is that your pacemaker kicks in at a level that's faster than your ventricular tachycardia, right? It does atrial pacing and can get you to break out. And when you break out, you continue to have a paced rhythm for a minute, and then you're in a new rhythm that's independent, that's stable again, and comes from a different node. So I actually thought about this because in the middle of all of this, my father-in-law, who has a pacemaker, was having episodes of ventricular tachycardia, and I was like, oh, That's bad, right? I'm an oncologist, that sounds as bad as it gets. And his cardiologist was not the least bit worried. And it's because he knew that his pacemaker will just pace him out every time. I'm like, okay. I hope there's a defibrillator in that pacemaker too. But nonetheless, I think it's a really good analogy for, when we're in a period of uncertainty, like we are now, how do you get yourself out? And it's by going back to basics, and not by going slower, but going faster but not crazy. Going faster but steady. And so that's what I'm going to talk about. How do we pace ourselves through this? So this is a model that I've been using as I think about how do we pace ourselves, okay? I think about 5 things that can help us walk through a space like this. And you would think that maybe as a scientist or as a physician, it would be very, very science-focused, but it's not. All of these things, though, I think are relevant to all of us as physicians and scientists. The first one is tell our story. That one's going to be obvious, and I'll explain in a minute. But we can't just tell our story to ourselves. We need to tell our story in different audiences and really engage with communities in different ways. I think we need to learn to support our trainees and bring up our early career investigators in different ways than we have in the past, and double down on creativity and innovation. And then finally, really working together, uniting in common themes. So just to walk through these. So let's just start with telling stories. So I'll tell you a story, okay? So, but maybe not from right there. I'll stand back. Okay. So I specialized in kidney cancer, as you've heard, and that meant, in addition to metastatic kidney cancer as a medical oncologist, I took in patients who had hereditary risk for kidney cancer. So von Hippel-Lindau syndrome, they carry one mutated gene for VHL and have very high risk for getting kidney tumors, but also tumors in their cerebellum, their spine, their retinas, their pancreas, and their adrenal gland, okay? So they need somebody who can quarterback all those things. And so over the first decade or so of my career, I worked with a group that developed a set of guidelines to be able to help these patients navigate this complex risk, which basically meant how often do you need to go do scans? Can you get away with every 2 years? Do you have to do them every year? We kind of came up with algorithms and then made sure they were seen by the doctors they needed to be seen. I'd see my patient, we'd have all their scans, we'd go through results, they'd say, all right, this year you're good to go, see you next year. Or we'd say, no, this year you got to see the neurologist or the neurosurgeon or the ophthalmologist. And, but the effect of that process was that we were able to really extend survival and prevent unnecessary surgeries so we could do them at the right time. Okay. That's satisfying, but not that satisfying. What you'd really love it is if some of the science from the laboratory led to the discovery of a key driver. I already told you about a cool key driver called HIF. And the development of maybe a drug that could actually target HIF, and the development of an oral drug that you could take once a day that would inhibit this protein called HIF2. And so the development of this drug was actually taken to patients who have von Hippel-Lindau disease rather than patients with widely metastatic disease who are late line, and offered to these patients, and the goal was to prevent further surgeries. Okay, so I had 3 patients that I'm going to tell you about. It's a family. When you take care of VHL disease, they all come in families. So it was mom, 2 daughters. Daughters were 18, 19 when I met them. They were lovely and very clear on they were never going to have families because they did not want to transmit the VHL gene any further. They were happy with what we were doing, but they were not going to go to college. They were not going to, they had made a lot of decisions about what their life was going to be like based on an assumption of their life expectancy and their life quality. So this drug comes along, and nobody knows really what the side effects are going to be like. Nobody really knows if it's really going to work. They know what they have to predict, but mom went on. She was doing okay. Daughter number 2 went on. She was doing okay. Daughter number 3 went on. And for those of you who know the story of belzutifan, all of the patients on this study saw their tumors stop growing and gradually regress. And what it meant was that people who would have, on average, 1 procedure a year went to none. Complete game changer with a drug that you take once a day. So that is not actually the end of the story. The end of the story is about 2 years on the drug. Both daughters are now 24, 25, and they came to me and said, we want to go off drug. And do you know why they wanted to go off drug? They wanted to go off drug because they were both getting married, because they both were ready to start their families, and they both knew from our experience before how much time they had before they were likely going to need a surgery again, or before they'd need to go back on drug. And they both had sat down and calculated that they had at least 3 years, and they were good to go. And they have done very well. And that's, the ownership to the patient, the ability to have something to offer someone that can change an entire family's outlook. I mean, that is exactly why we do what we do. So I really encourage you to, at the time I didn't think anything of it. These are my patients, like you see them one at a time. As you experience, interactions with patients make good stories, but also interactions with, mitochondrial assays and others also make for really good stories. And, think about what stories you have that you can tell. All right, now the hard part is, who are we telling these stories to? And how do we find an audience to tell these stories? So my challenge is to help us think about how we share these stories in dedicated sessions or places where we can balance the storytelling with listening to other people's stories. Because that is where we're going to get real dialogue in the community. I think in doing that, we have to be able to show, I'm talking about people in the community, we have to show our passion, and that's also showing our commitment to the work that we do and sharing together our time and our talent. I have a couple of examples, because I think this is the hardest one, because I think people come away from this thinking, do you mean I'm supposed to talk to everyone on the airplane? I do, actually. That's even hard for me, but I do it now. And it always goes okay. I wouldn't say it's always great, but it always goes okay. One of my favorite stories was actually, I moved out of the NCI. I had a whole bunch of boxes, and I had to move them, and so I took them to Staples to ship. And the guy was like, why do you have so many boxes? And I was like, I've been working on cancer here, and I'm moving out, and I have to do something different with cancer in my life. And he was like, what do you mean? And so we talked about cancer for like 5 minutes. Not a big deal, but somehow we ended up talking about lung cancer screening and the importance of doing that. You never know how these conversations go. But that was not all my boxes. So I came back 3 days later with more boxes. And I came up to the counter, and the same guy was there, but a bunch of people came from the rest of the store, and they were like, you're the cancer lady. Like, we want to talk about cancer. So you just never know where things will come. But some opportunities are built for us. So I want to tell you about one, which is Pelotonia. So Pelotonia is a bicycle event that's very tied to the Ohio State University James Cancer Hospital. And it's a local event. And we have people in town who ride and support, do fundraising to support the cancer hospital. But it does a bunch of different things besides earn us enough money to be flexible, right? $300 million over the last however many, 15 years, currently is in the range of $25 to $30 million a year. So real money that comes from regular people who go out and ride their bike, 25, 50, or 100 miles. I only did 25. But it also creates an opportunity when you're out on the bike to talk to people about their cancer experience and why are they interested in cancer research? What do they care about? But more importantly than that is that it also provides an opportunity for us to really engage with the community, because Pelotonia works year-round to garner community support by tapping our faculty and our scientists and asking them to go out to speak to people in the community. They actually pay to provide some media training, some storytelling training to our faculty so that that is a more comfortable and productive conversation. So these kinds of things are, I think, where across the nation we need to figure out where to find those positive venues. It also provides a lot of support. It supports our scholars, it supports our recruitment packages, it supports other initiatives that we do. We can also try to find ways to help our community by really thinking about what they need. Like you, I come from a state with a lot of rural counties. Actually, I was really surprised. I heard you have 87 counties, we have 88, so really, really similar kind of things that we have to think about every day. And in our catchment area, or in our state, when we've asked about what are some of the biggest needs, we have whole counties that have no primary care, right? And regions that have no access to advanced imaging. We've had a mobile mammography unit for some time, but this year we put on the road a mobile low-dose CT for lung cancer screening. And it's widely accepted and great uptake. There's a lot of criticisms of these kinds of approaches that you'll see in the media, suggesting that we're going out and doing scans and then letting people figure out what to do with that. We are not. When we go out to the community and schedule a patient for a lung cancer CT screen, we keep their information, we make sure that they land appropriately when we find anything that's concerning, and if we know that they're fine, we give them a good follow-up plan. Because people really deserve, whether they're far away or near to an academic center, to be able to control their own health. Just like my patients were looking for with von Hippel-Lindau syndrome, someone who has high risk for lung cancer also deserves that ability to be able to understand and know their risk. So we're out on the road. We're engaging with communities as well to try to educate on prevention, detection, and treatment, really trying to develop more relationships across Central Ohio and working with our healthcare providers. We also find that this increases enrollment to clinical trials. Being able to talk openly about the value of clinical trials is a huge asset. And I'll also just put some data up, and there's kind of too much data on this slide. But what this data shows, this is from the AACR, just published in September. Because they asked, is the public actually not interested in us doing cancer research? Are they actually wanting less cancer research? And the answer is that actually the public is very interested in cancer research. So the graph here on the left shows, although there's been some decline in how many express a great deal or fair amount of trust in scientists, it's still the overwhelming majority of people who trust scientists, either a great deal or a fair amount. We can also show, we'll go to the lower right-hand corner for just a minute, increasing support for federal funding for research. You can see there it's 83% say yes, strongly favor or somewhat favor increasing federal support for research. So this, I think, helps a lot when you go out and know that this is a receptive audience. It's a savvy audience as well that has some opinions about what we're bringing to the table. In the top middle. So scientists are seen as intelligent, 89%, okay, good. But honest, 65%, okay. I think we try to be honest, but sometimes we sugarcoat. I mean, we have to be like honest, right? Feel superior to others, 47%. And good communicators, 45. Closed-minded, 28. So whether those are correct or not, that's the perception. And so we can only go into an audience knowing how we might be perceived. And that can help us be more transparent, more honest, more able to connect with someone in a way that feels more authentic, because that's more of what people are looking for. And then finally, I think this piece of data, which is maybe the most concerning, is how many Americans can't name a living scientist? Okay, 72%. So, if we at least go out and talk to people on airplanes, and when we're standing in line at the grocery store, they'll know us, right? And they can say, I know that one, but who are they naming? It's Tony Fauci, Neil deGrasse Tyson, Bill Nye, science guy. So this is important that we find ourselves in the community so that we can help grow the appreciation for research, the important research that we do. And then this is, I think, the most important slide, although there's a lot of more words on here than there should be, is how do we really embrace this? Two-way engagement. So looking for places where we can listen as much as we talk, and finding ways to show the real world need of our research. For everyone's research, I mean, I do very basic research, but there's a real world need in understanding how cells make decisions about what their metabolism is, because in fact we all eat, because we all have metabolic components to our makeup, and because there are drugs that affect all of those things, and more that we could make where we can have control. I think remember things that shift people's minds, which is not more data, okay? It's really, I've learned this one the hard way because I have a tendency to try to give more data, okay? It's not more data. It's how competent do we seem? Do we know what we're talking about? The integrity of our words and our actions. Benevolence. You know, are we doing this to benefit others? And our openness. You know, how much do we want to engage as much as share? And then just a few suggestions about how to find non-traditional venues for making some of these interactions. I just want to say, recognizing partners as real partners and real partnerships with groups that are already ready and out there who really, really want to engage, like advocacy organizations. That is your, the sweet spot. That is groups of people who care very deeply about the work that you're doing. But community services and anything that can create good venues for listening. And so, when I think about this phase shift that we're living in, we can be the role models that help to change. And then I just put this up there to remind us, right? Because I think there's a tendency, like, let's put it out on social media, okay? When you put something on social media, the people who see it is all the same people. It's not all the people, and it's not our field that's the lone. I create content about content marketing for content marketers, marketing to other content marketers, right? I talk about science to other scientists so that they can do more science. But we can break out and share it with others as well. Okay, that's my preachy one. The rest are good and easy and obvious. Right? Supporting trainees and early career investigators. Okay, I already had them all raise their hands, so you know who they are in the audience. So first of all, this is not a new thing that it's important that we get early career folks moving toward independence a little bit quicker than we have in the past. This graph, which you can see, dates back to 1980, shows the shift from the first R01 at age less than 35. You can see that used to be 20% of people got their first grant before they were 35, and now it's pretty darn close to zero. Okay, and in the meantime, holding a grant, an R01, and being over age 66 is now just climbing and climbing. And so this is an area that's really worth paying attention to. How long does it take in graduate school? What do we need from a postdoc? What's the expectation of an early career faculty member? How can we enable people's ideas to get out there and be in the real world in a more timely fashion so that we have the time for people to be most productive? I heard a really interesting anecdote at a meeting that I was at a couple of weeks ago. And it was about a famous physician scientist that many of you may know, so you might actually know this story, John Minna, who was apparently a division director at age 35, okay? I mean, think about that. Why do we all have to be 50 before we do anything? So I think we really can identify talent, ripen it fast, and allow people to really sit at the table with us. So I think we can talk about all the different ways that this takes shape, from everything from pre-high school to high school. Here I just have a couple of examples of stimulating that interest from interest groups in medical school, stimulating interest in things like radiation oncology. Our spin doctors is a medical school group. So we're really trying to support both the pipeline of physician scientists, but also the recognition among, in this example, medical students that science is for everybody. We, at least in cancer, I don't know of any place where we're done, where we've got all the answers, where all of the treatments are as good as they could ever be. Okay, there is room for more. So there is room to learn and push boundaries much, much further back, and all of us can play a role in that. And so I think engaging medical students as well as earlier undergraduates to come into the research field is absolutely important. There's just another couple of examples of how we do that. And preparing people to lead. This is sort of to what I was suggesting before about thinking, about how we bring people up a little bit more quickly. Thinking about at each of these stages, how do we enable our young people to be, full members of the team. So, and turning to these young people as valued partners, I think why should we do this? Think about who's best able to flex if we're in a radically different environment. Okay, it's people who are bringing a different kind of experience and who are coming at this without some of the baggage that we older folks have. Who understands the changing landscape of social media? It's not me, that's for sure. Who's grown up with ideals of diversity and thought as a standard practice, and who benefits most from today's discoveries as advances in healthcare that will be for us for 20 years from now? And who can relate most effectively to communities that are outside of science as well? I think in a pacing model, it always is worthwhile to say, let's double down on creativity and innovation. Let me just give you an example of what that means, because I think it's easy to say and hard to do, because we all are doing innovative work. That's what makes us tick. But really thinking outside the box and thinking, what could we do? What's the realm of the possible, right? So I just want to give you an example of a trial that we're doing at Ohio State that I'm so excited about. So this is a trial for a very rare subset of pancreatic cancer. So pancreatic cancer is not that common to begin with, sort of in the middle of common. But if you're an FGFR mutant pancreatic cancer, that's rare. Okay, there are FGFR inhibitors out there, oral drugs, tested in other disease states, and you might have the bright idea that you do a clinical trial. Just a nice phase 2 clinical trial in FGFR mutant pancreatic cancer. Okay, we could wait for decades at Ohio State, where we see lots and lots of patients, to see enough patients to fill up this trial. With all the advertising in the world, it's just not that common. And so the brilliant idea that one of our groups had was to identify these patients not by waiting for them to come, but by reaching out to the groups that actually do FGFR testing, right? This is all commercial. And so finding the commercial sponsors that identify these patients and ask them if they'd like to be contacted by Ohio State about a possible clinical trial. So if they're eligible for this clinical trial based on their FGFR mutation status, and they talk to us and would like to participate, then we engage their doctor locally and say, if this is the right time for your patient to consider something like an FGFR inhibitor, just let us know. And then we talk to them by telehealth. They're our patient, and we ship the drug to them to their home. They take the drug orally. They see their local doctor for their labs, for their physical exams, and they have to upload their scans, okay? Which is all doable in 2025. And I don't know the results of this study yet, but it's accruing really well, and it's accruing all over the nation to allow patients to have access to a drug that otherwise it would be prohibitively expensive to try to offer much more broadly. So really excited to think outside the box, really, really be innovative. I like to think about the things that we do at Ohio State, and you have these lists as well, sort of things that are first, best, only. First in human, we have a new phase 1 drug, fully developed from its original concept from the Ohio State University. Tests dihydroorotate dehydrogenase and as a novel pathway inhibitor. I already told you about our 100% telehealth clinical trial, another novel GARP CAR-T that we're working with as well. So I think being able to push the boundaries from the discoveries that we make in the laboratory and really translate them and make these things available for patients is some of the ways that we can really push the envelope. You can't talk about innovation and pushing discovery without talking about AI. So just a handful of examples of things that we're doing at the James, looking at biomarkers for precision treatment of head and neck cancer, validating diagnosis in breast cancer, enhancing human analysis speed and accuracy for pancreatic cysts, and predicting cancer severity and whether chemotherapy is even needed for patients with colon cancer. So a variety of things that use the electronic medical record, that use the histology of the patient's biopsy, or use the imaging, or all of it, all of the above, to make accurate and helpful predictions. So we are finding this to be a real game changer, as it is everywhere. And then in the space of clinical trials, we're also trying to double down a bit on pragmatic clinical trials, really focusing on essential endpoints, the things we really need to know, cutting out unnecessary data collection. We're trying to use virtual staff support. When I was at the NCI, we did a pilot as well with virtual clinical trials office, right? Why does the data coordinator have to sit in the office of the doctor? They're actually tied to the study. And the work that they can do is actually very well suited to virtual interaction with the EMR. Same thing for some of the nursing, research nurse duties to interact with the patient for consenting, for answering questions, for some of the follow-ups as well. That can be done, especially as we're looking at more and more diversified clinical trials that are going to be happening in far too many sites to have humans, to centralize the humans, but distribute the clinical trials. Broadening access and enhancing reach, partnering with government, industry, and academia, and including the beneficiaries in their research. So I think that this kind of pacing can lead to a virtuous cycle of asking bold questions, courage to push those boundaries, think out of the box, flexibility in approach, and create new models for collaboration. And then the fifth is, I think should go without saying, but that we're always stronger together. And so, the analogy of the single twig compared to the bundle is suddenly unbreakable. And this has been happening for the past several years, but really is happening in great ways, I think, across the cancer centers, especially leaning into the cancer centers as a network. So I'm delighted to be here because I think Ohio State and Minnesota should be working really closely together because we have so many areas of alignment and areas where we are tackling many, many of the same problems. University and institutional cooperation versus the competition that can sometimes be rife, expanding partnerships with foundations, nonprofits, and industry, blurring boundaries. I think about the natural sciences and liberal arts, for example, and acknowledging gaps and building reliances. And then finally, really confronting the economic realities together can really help us as we move forward. So that's my framework. And to me, they all tie together. We're encouraging everyone who's a part of this to tell the incredible story that is the story that makes up what it is to be doing what we do in cancer research. To engage with our community as a real partner and not our client, but a part of this whole amazing story, doubling down on how we engage our early career members and supporting them as full valued members, asking bold questions that challenge norms, think outside the box and approach with flexibility, and uniting in common themes so that we can accelerate discovery, effectiveness, and change together. And with all this framework, as I was putting it all together, I realized we've actually been here before. About 25 years ago, Paul Calabrese published this paper in, now I lost the reference. I can't remember where. But what you can see is that, in fact, this model is the model that we all have been trying to work from. That it's one wheel between healthcare providers, government, and private organizations with all the components in between, so that we really can make a big difference together. So with that, I thank you very much for your attention, and I will be very glad to take any questions.
Before questions,
or not.
Thank you very much. We have a special award for you. If I can get it the right direction.
Thank
and
you.
then you have to smile for the picture.
Okay, yeah, come on. Yeah. All right, thanks.
So Kim, one thing that's very clear to me is now we know why you were the director of the National Cancer Institute based on the quality of this presentation today. So thank you.
Thank you.
And I think we're going to open it up for questions. And we have a microphone here to pass around for anybody who wants to grab it.
So that graph you showed of R01 grants going towards investigators over 66 versus those under 35 was quite shocking and trending in absolutely the wrong direction. What do you think is the basis for that? And I would argue that getting those awards over 66 would be completely unable to do so without those investigators under 35 years of age.
Yeah. So I think there's a lot of factors for sure. If we just look at students we see applying for graduate school, like it's become very normal to have to do a couple of years or 5 years of post-bac type experience or laboratory technician work. And then postdocs have gotten long, and then we add an instructorship, maybe 2 postdocs because stuff is complicated. If you're really a top-end postdoc, you're trying to get your paper in Cell Science and Nature, that's a year-long process at the best, right? Like there are lots of things that are factoring into pushing this stuff really out. And having been, department chair and now in my role, looking at, hiring, like, does everybody have to have a K99 or a K08, or can we take some risks and bring some people in and support them to it? I also think that K can be a hindrance, although I love K awards very much, because you can't write an R01 while you're on a K. And I saw far too many people when I was a department chair because we did try to bring people in without the K, right? And then they wouldn't get the K for 2 years, and then that's 5 more years, so they're not getting their R01 until like year 6 or year 7. It's really, I think that part is pushing things out on the other end. I think it's not surprising that I think 66 is sort of the new 55, right? And so that's a big group of our investigators who are very successful and know how to write grants. And I think your point is very, very correct and spot on. I think we need mechanisms that allow for more collaborative grants. I think we should get rid of you have to write your first R01 independently and you can't be a co-PI. Wouldn't it be lovely if your first R01, sorry, I was the NCWI director, I tried to fix it. I was not there long enough. If your first R01 could be with a very senior, or you're a senior investigator, and then go on from there. I really think we could do this a whole lot better.
So there's pretty exciting ideas, but in addition, how do we encourage people who aren't working in a lab in finding some new gene regulating tool or new piece of a drug that in the future is going to do something? What about people who will understand how to be the cancer communicators and work with the community and develop what the community needs, and the storytellers who still have to count? They can't just be publicity hounds.
Yep.
They have to be able to tell how the science is going to affect patients and get it into the community so people will be eager to participate and get brand new therapy. How do we recognize those skills at the university level?
Yeah, I think I've been really pleased to see things like implementation science be, fundable work, because our work isn't done until it actually gets its way out into the hands of patients, into physicians who actually use the therapy or the output. I think that we have to value a lot of that work. I mean, this is where COE came from for the CCSG, was it didn't provide a lot of money for it. I know that too, but we have to try to find ways that we can provide that kind of support, because in order to have somebody who can have that kind of dialogue in the community, they have to have time. So where does that come from? Philanthropy helps a lot in that regard. I think partnerships that allow for in-kind type of work can also help a lot when we're thinking about people who are outside our academic community but who can also play that kind of role. There are other ways to get that. It's expensive. I think. And you're raising a really good point. Like, how do you buy people's time to go out and do storytelling? I think it's worth the time to send them out there.
How do you get deans and cancer center director and department chair to recognize that that's an important process, that's a legitimate faculty activity when they're not taking care of patients?
Yeah. So I think if you can show that it's bringing more patients in, I mean, there is ROI to come from it. I mean, you asked me this as a CEO. I mean, I am putting effort into this at the Ohio State, right? We have 10 investigators every quarter who are going through storytelling training. That's like 20 hours of training. And then they will be the ones who are going to be tapped to go out into the community. So it comes out of their work week. And we are creating that time. So how do I convince other deans to do it? Because I also have lived in, like, the bottom line is as low as it can be, right? And so there, I think we really have to, be really judicious about who we're sending and when, and finding the people who have that time to spare. I think this might be a really great opportunity for more of our senior faculty as well, right? Who might be more nimble in the kinds of work that they're able to do, because writing a grant is a lot easier when you're writing your 10th grant than you're writing your first grant. But I think we can just, kind of work with department chairs and people who know their faculty to know what we can and can't do. Not easy. I'm not going to try to pretend it's easy, but we have to have those conversations.
Hi, thank you. I was wondering if you had any advice for the young career scientists who are kind of just starting out, and what they can do to help prepare for their role moving forward.
Oh, yeah, I don't want to let go of it. It's so.
No.
I'm going to get it all fingerprinty. So the first thing that I would say is really be aware that this is the best time ever to be in cancer research. The kinds of things that we can do, I mean, they're just phenomenal. So it's easy to kind of think like, oh, this is really hard, but it's actually really, really cool and really, really fun. So I would start with that. But I think in terms of like really preparing for the workforce is, we have to know how to be resilient, right? This kind of turbulence has happened before. This is kind of like a lot of waves at once, but this has happened before, and we all have learned how to be resilient. So working with your mentors on how to do that. But I think also, being kind of committed to timeframes, trying to make sure you're getting grants and getting papers out so that, knowing that kind of slope of that curve, so that you can be one of the ones who's writing an R01 before you're 35. Because there are milestones that we all just have to make, have to finish grad school, have to finish postdoc. It comes with life, like, I had a kid in grad school and a kid in residency, so that all factors in, but figuring out where you can be, on top of getting your papers out the door, and on top of getting your grants out this cycle, not next cycle, and being very in touch with all of your mentors. I would say those are my top things.
Thanks for this terrific presentation. So I have a question for you about kind of funding. We know funding is really low right now. It looks like Ohio State relies a lot on philanthropy, which I think is one way to really generate extra resources for especially pilot funds, offsetting some of these activities we're talking about. But I'm curious your thoughts about other really strategic ways to offset. I mean, we're talking about clinical trials that especially cooperative groups are underfunded.
Yeah,
we're talking about it taking longer and longer for people to get their own grant funding, which they're needing to be supported at that same time. And so I'm wondering if you have some thoughts about that.
Yeah, I mean, I think every scenario is individual, is the first, so that I don't have a blanket answer that works for everyone. But I think being really open to looking for clinical trials, right? Looking at the portfolio of trials, at Ohio State, we really like IITs. We have a lot of IITs. IITs are not super well funded. I think we have to look at, do we need to balance in some industry-sponsored, really good phase 3s that move the needle? And interestingly, we push the companies on that, and they're like, I said, we want to do that. They want us to do that. but we want some credit, right? We want to have, some good position on the paper, or, some teed up for the next one. And they're actually very willing to have those conversations. So I think thinking about that portfolio so that we have a balance of things that are financially underfunded and some things that support the program a little bit better is one way to think about it. I think really looking at foundations and being very judicious about foundations and making sure that they are well aware of the work that you're up to and doing, being very close to philanthropy. I mean, all of these things are really going to be important when funding gets really, really squeezed. Oh, and I think the other thing that I'll say is making sure our grants are really just tight. So I think for young people in the audience is, there can sometimes be a barrier to show someone else your grant, or someone might see that your grant isn't as good as it might be. That's the person you want to have tell you that, not your study section, right? So like leaning into mock reviews if you do those, or other ways of ensuring that every grant has the best possible story that it's telling so that it has its best chance.
So thank you for that really great talk. And I'm going to basically say what she said, but go up one level on the elevator. So there's a huge economic imbalance out there, and there's this addiction, right? So in my own story, we had a really great collaboration with an industrial partner, and we had a project that worked from the back of a napkin onto human clinical trials, and we were successful. And the year we were successful, the money disappeared because the project was done. They didn't need us anymore. Now it was out there.
Yep.
And all that time we had, gobs of money that we were using for the project, and all of a sudden you face the money's not there, and you still got all these people that you have to pay. I think the same is true for indirect costs and salary savings, that institutions have basically gotten addicted to this. And all of a sudden, the administration wants to cold turkey it out for whatever reasons, right? Political or otherwise. So how can we improve the economic balance? There's tremendous amounts of money tied in industry with very little innovation. Like, do we really need 700 anti-PD-1 antibodies? And there's very little innovation going out there. So in your position as a former NCI director, can you think of ways to actually influence the powers that hold the money to make this a more productive economy? Not a meritocracy or handout, but really just a more productive economy.
Yeah, I think the thing that I'll say is that stability goes a whole long way. So wherever it's possible, and that's the whole problem with where we are right now. Like, things are not stable. And what you just described is exactly, those cliffs, which, in other sectors are normal. Like that to have a big team and then suddenly have a really small team. But we're not built for that. And the kinds of ideas that we're usually working on sometimes take much longer than what our funding models kind of allow for. So I think where the government can create some stability, it's proven over and over again that it's actually really valuable. So example that I'll give is the R37 for early career investigators, right? So early career investigators who apply for an R01 and beat the pay line can apply for this R37 and have 7 years of funding. And so we tracked people who were just above and people who were just below the pay line. And how did they do on getting their next R01? And it's like 2 to 1. And we all know that the peer review process is not that precise to be able to pick winners and losers. But having 2 extra years definitely gets people to the second R01 better than having 5 years of funding, right? So I am a big believer in how we try to create some of this stability. I think we can try to influence some of the government funding models to try to create more stability. I think there are some people who will find that to resonate. I think we're very fortunate to have a new NCI director who lived on R01 funding his entire career, definitely lived through peaks and valleys. He gets it. I think he will resonate when the government's open and we can all talk to him again, with the idea that opportunities for funding that are more stable and support the ongoingness of projects will go a long ways.
I really enjoyed your talk. One of the things that is really pressing to me is the attack on DEI and our T-32s, which in the past I would write so much about our recruitment efforts.
Yeah.
When you talk about messaging,
Yeah.
messaging in Cleveland, your messaging in Southeast Ohio, how do we expect to have that messaging if we don't have scientists and researchers?
Yeah, who represent the people? Yeah, yeah.
Who trusted to communicate with these populations?
Yeah, I agree. I think it's important. I mean, there's good data that people trust individuals who come from a different or the same cultural background. So I think it is still important that we identify the best talent that represents all of America, and America is diverse. So I don't have a good explanation for how we kind of navigate this abrupt transition that's happened in sources of support that help to raise that awareness, but it's still very much a part of how we're going to be successful.
Do you have any advice? And I'm looking at some of my lab people in the audience. So peer review has gotten to a very funny level. You convince us that we don't have all the answers and there's still science to be done. But peer review, the bar for getting things in good academic journals is getting, I think, more complicated than it was 10 years ago.
Yep.
Any insight or experience to?
Yeah, I have one
give us some
message that I give
question?
on this topic. I think this is true for manuscripts and grants. And this one is the hardest one because the problem is us, right? Like we are the peer reviewers. Okay, I'm just gonna say it again, like we are the peer reviewers. So, if I had one thing that I could go out on the road and try to like preach to the converted, it would be like, we all need to kind of own what it is to do good peer review. And that is when we're asked to review a manuscript, to read it and provide this is also me speaking as a former editor. I would have loved this more. A crisp overview of the salient strengths and weaknesses, and does it fit the journal and its scope? Like that's all you really want to know. I don't really care about like whether the stats are all right on figure 2, okay? If the stats are totally bad, that's a critical weakness. The stats are flawed throughout this entire manuscript, and here's an example of why. But the nitpicky detail all the way down to, you used in the grants process, you're using, the wrong pH buffer because I know that pH doesn't really work. Please, let's not do that to each other anymore. I think we can look, and I think actually of all the things that have changed really recently is, and I have not done peer review, but my husband recently did, so I got to hear his like on the side version of what peer review is looking like. Has anybody else done a recent? Yeah, so you can tell me if this is wrong, but my sense is that it's more on the big picture of the topic and on the significance and the overall impact. And in some ways, that's good, because I think that moves somewhat away from, trying to be able to discriminate based on some little nuance that really isn't in the big picture scope. So. I think I'm going to keep going on this one because this is a soapbox that I talk about. Because we have a bunch of students and postdocs in the audience, I would also say you're going to be asked to review papers for your PI, and I would read the paper and ask your PI to talk about the paper and to think about the paper. Here's where we can use AI to our advantage. I mean, you can load it in and listen to it as a podcast if you want, right? And you can get a better sense of like, what is it that this paper is trying to tell us? Does the data actually show that? And is this important to the level of whatever this journal is? Because if you're reviewing for Nature, it should be different than if you're reviewing for Oncogene. And I publish a lot in Oncogene, so I'll put it here actually. But as you all know, it doesn't matter what you put out there. Our critiques right now are really way too much.
We need one more question because Congresswoman Kelly Morrison is on her way.
Oh, no.
So, anybody else?
So apropos the comments you just made about manuscripts, one thing that's happened, as we all know in the last, 10, 15 years, is the amount of information and detail of some of these manuscripts in Cell, et cetera. I don't know how any reviewer, I don't know how any author can get through all that information.
Yep.
And with 8 figures, each with 20 subunits. So are we just expected to kind of trust all that and look at the big picture?
Yeah. No, I personally love papers that are simple. And yeah. I don't have a good answer for that. It's true. It's gotten out of control. And I think the peer review process, when we ask for a bunch more, because it would be interesting. I always found this as an editor, like I wanted to cut people back, and I did this sometimes. Because they would say, it would be really fascinating if you made this mouse model. Well, you know what they're going to do? They're going to go make that mouse model. But we don't have to be fascinated. And they can actually just stop with a really good story because there'll be another one. And so, yeah, maybe in my next life I need to be a journal editor, and I'll be like, here's how many figures you get, just a few, yeah.
I just wanted to say that I totally agree with what you were saying about manuscripts. I've been an editor of 2 different journals, and I tell people, I don't know what happened during COVID, but everyone got nasty.
Yes.
Did you notice that?
I agree.
Even on the road rage, I mean, everyone just got into this complain mode. And so what I tell my reviewers is, you need to mentor the authors. You need to be constructive. If you have a problem, and then you need to, instead of just complaining about the paper, you complain about the paper, but then say, you need to do this. What do you do about it? Like, what can they do about it in 2 months instead of 2 years, right?
Exactly.
If they go home and make a mouse, that's going to take a couple of years, right?
Yeah.
So, you have to pace you have to match the critique to the journal and also to the time frame, and be a mentor to them. Be constructive instead of being mean, right? So. Yeah,
I love that. I'm going to use that. I think that's beautiful because it is true. You're being asked because you're an expert. And you're being asked to review, and so why not think about that as a way of creating that more constructive type of review. And I think also trying to find ways that it isn't 2 years. I mean, that story that I showed a snippet of, that story went out for review. It came back with 16 pages of things to do and a year to do it in. Okay. In the meantime, like I've moved, Jeff's moved, the postdoc's doing something else, right? And I'm telling the postdoc, like, we got to get it done, and she's like, we got a year. This is like don't let it we need to just be done. I think if reviewers can help us by, just really trying to help us make the paper better, or tell us why it is that it doesn't belong in that journal, because it's okay. It'll find another home.
Great. Thank you, Dr. Rathmell. We're really grateful for your leadership and dedication to advancing science in the practice of cancer care. I'm going to go home and work on my storytelling.
Okay,
so
good.
Thank you very much.
Nice.
So before we adjourn to the refreshments and close today's program, I'm really honored to welcome Congresswoman Kelly Morrison, someone who shares the values championed by Dr. Kersey and Dr. Rathmell. As both a physician and policymaker, she's been a consistent advocate for biomedical research, healthcare innovation, and equitable access to care. Her leadership continues to strengthen the vital connection between science and policy. So without further ado, our final part of the program today is to welcome Congresswoman Kelly Morrison. Thank you very much. Thank you.
Oh, it's so nice to meet you.
Good afternoon, everyone. I'm standing between you and snacks, so I'm going to be brief. I appreciate that kind introduction, Dr. Miller. What an honor to be in this room with all of you and to share this stage with these incredible leaders and, of course, Dr. Rathmell. Quite humbling, to say the least. Dr. Rathmell, thank you for sharing your experience and for reminding us about the critical role that medicine and science play in our world. You're an inspiration to us all, and I know your remarks will stay with everyone here going forward. I'm Kelly Morrison. I have the honor of representing Minnesota's 3rd District in Congress. I'm the newest member of Minnesota's congressional delegation. Actually, I was sworn into office in January, and let me tell you, it's been quite a 10 months. I'm also an OBGYN, and I had the privilege of caring for Minnesotans for more than 20 years. And that work is one of the primary reasons that I first ran for public office. As an OBGYN, of course, I had always been at least adjacent to politics because women's health is politicized. But I had never considered running for office myself. I was busy raising my 3 kids, taking care of my patients, and trying to keep my head above water, if I'm being honest. But the promises that the winner of that 2016 presidential campaign made about limiting access to reproductive health care shook me to my core. I knew what it would mean for people across the country, and I was just compelled to get more involved. I had to do something. For years, I'd been concerned about the growing skepticism about science and expertise in our politics and our culture, and I wanted to be a voice for science and evidence-based policymaking. And I also really wanted to work to try to heal our divides. And there are ways in which I think physicians are well suited to at least endeavor to do so. We, of course, work to heal our patients, and we must learn to put ourselves in our patients' shoes to best understand and help them. We must empathize with them. And this moment requires that we emphasize empathy, in my view. We must empathize with each other with urgency right now. So a little bit more about my journey, and then I want to both thank and challenge all of you. I served in the state legislature for 6 years before running for Congress, and I passed more than 90 bills into law, many with bipartisan authors. I was a member of the first Doctors Caucus in the Minnesota legislature, which was bipartisan, and I helped lead the effort to protect and expand access to reproductive health care here in Minnesota, a needed protection after the Dobbs decision. Knowing how critically important the university is to our state, I was very proud to carry the legislation that provided much of the funding to renovate the chemistry undergraduate teaching laboratories at Fraser Hall. And I served on the Rare Disease Advisory Council during my time in the legislature. I think it's incredibly important that we have more elected leaders with scientific and medical backgrounds at every level of government. As the complexity of our challenges increases, we need leaders who understand and who have grappled with those complexities and can bring their expertise to be active participants in problem solving. So here, I feel compelled to put in a shameless plug. If any of you have ever considered running for elected office at any level of government, I would be more than happy to talk to you about my experience and how I can help in any way. Regardless of your political affiliation, we need good people in government. If you've ever thought about it, please do it. I mean that. We're living in difficult, challenging, and frankly historic times, and we need all of our best people to step up. In what has been a pretty dark time for science and academic freedom, I do want to share one bright spot in Congress, I think, right now. There's finally a critical mass of doctors. So we formed the first ever Democratic Doctors Caucus in Congress. There are 6 of us. We represent a variety of specialties, and we come from all corners of the country. And one of the first things we did was call a press conference to explain our opposition to the nomination of RFK Jr. to lead the Department of Health and Human Services. He has, I thought I might get some response from that one in this room. He has no medical or scientific background, has never run a large organization, and has profited off of spreading conspiracy theories and misinformation about vaccines, mental health care, and reproductive health care. And of course, our worst fears began to be realized almost immediately after the Senate confirmed him. From the get-go, he began to dismantle our scientific and public health infrastructure and continued his habit of undermining the integrity of long-standing vaccine policy, now from a perch of significant power and influence. The cuts that he and this administration have made to our world-renowned public health institutions and research universities have been shocking. Institutions like NCI, NIH, CDC, losing billions of dollars and a generation of talent is short-sighted at best and threatens both the health and well-being of Americans as well as the leadership role of the United States in medical and scientific research and innovation at worst. The funding cuts and worries about the chilling effect the attacks on academic freedom will have on critical transformations in patient care, cancer and rare disease research, and to the overall health of our nation are frightening and honestly just enervating, especially to those working directly in those spaces. You are all leading this work in a profoundly difficult time. At a time when it can be incredibly challenging to take care of patients and conduct research, the important work that you all are all doing when the future seems so uncertain, it can feel overwhelming. But I want to remind you of how important you and your work are. We need you now more than ever. This is a watershed moment in our nation's history. It truly is. We are witnessing a slide into autocracy that many Americans never thought was possible. But out of crisis can come opportunity. We need to lean into issues and endeavors that bring us together, and your work can be a unifying force. All of us want to be healthy. No one wants to receive a cancer diagnosis, or worse, a diagnosis without a cure. Your work helps people, and helping people brings them together. Your work can both cure disease and help heal the hyperpolarization that afflicts us. Because in the end, of course, we all have more in common than we don't. We all just want to lead healthy and thriving lives. So please keep going. So many Americans are so grateful to you and are cheering for you. We need you to continue to conduct research and continue to seek the next breakthrough or innovation. And I want to encourage you, actually implore you to add one more thing to your already full plates and get involved in advocacy. As I mentioned, most politicians don't have a background in science or medicine, so your voices are crucial. We need you to help inform them about your work and to help guide policymaking so that Americans can thrive. I encourage all of you to get involved in advocacy work and make sure that your elected officials, no matter what level of government, hear from you. Your voices are respected and your voices are powerful, and we need you to use them now more than ever. Here's a current small example of what I'm talking about. Advocacy from physicians and others with medical expertise help revive the Newborn Screening Saves Lives Reauthorization Act in Congress, which strengthens screening programs that detect life-threatening and life-altering health conditions immediately after birth. I'm leading on that bipartisan effort in large part because of the advocacy of experts who recognize the danger of cutting that program and brought those fears to leaders on both sides of the aisle, and we now have a good chance of protecting it. Another way to advocate is to share your stories. We were talking about storytelling. Tell me and my team how an NIH or an NCI grant made your work possible. Tell us about your experience caring for patients who benefited from a clinical trial. And tell us what you want to see for the future of cancer and other research in Minnesota and across the country. We can amplify those stories to help spread the word about the transformative power of the work that you do. I want to conclude by offering my heartfelt gratitude. Thank you for the work that you do. Thank you for sharing your talents with Minnesota, the United States, and the world. You and your work are the reason Minnesota has been a world leader in medical and scientific research. Your work transforms lives and your work saves lives. Please know that I will continue to do everything I can to protect academic freedom and Minnesota's leadership role in scientific and medical research. Thank you again for including me today. And thank you, each and every one of you, for the work that you do. Please stay in touch. Thank you so much.
So I would like to give one round of applause again for both speakers. I mean, that was very inspirational. I think, we need to live in these times with some level of hope. And as I've told many of you, I think it's going to be okay and to keep working. So thanks, everybody.